So back in May of 2024, a really interesting study came out, and it could totally change how we fight one of the biggest threats in modern medicine. So today we're going to break down exactly how scientists are engineering a brand new superbug killer, pretty much from scratch. You know, for nearly a century now, we've had this amazing safety net, antibiotics. But that era, well, it's coming to a really dangerous close. The very bacteria we've been fighting are, well, they're evolving, they're getting stronger, and they're basically learning to just shrug off our most powerful drugs. So this really begs the question, right? It's the kind of thing that keeps scientists up at night. And in this explainer, we're going to look at how one team of researchers decided to go back to the drawing board. But not just any drawing board, a digital one, to design a totally new weapon from the ground up. Okay, so to really get why this new approach is so revolutionary, we've got to first understand just how big this problem is. I mean, we're not just talking about a few stubborn germs here. We are facing a full-blown global crisis. This is what's called antimicrobial resistance, or AMR for short. Basically, it's what happens when we expose these microbes to our drugs over and over again. They learn. They develop defenses, and eventually our treatments just stop working. And this isn't some fringe issue. The World Health Organization has flagged this as one of the top public health threats on the entire planet. So the researchers in this study decided to go after two of the most notorious culprits out there. On one hand, you've got MRSA, which you might know as a really tough staph infection. And on the other, E. coli. Now, what makes E. coli especially tricky is that it's what we call a gram-negative bacterium. Think of it this way. It has this extra super tough outer membrane that's basically like a suit of armor, and it makes it incredibly hard for our drugs to actually get inside and do their job. All right, so if the weapons we've built are starting to fail, where do you even begin to look for new ones? Well, scientists are turning to a kind of surprising place for inspiration, nature itself. It turns out the world is absolutely full of these things called antimicrobial peptides, or AMPs. Our own bodies, plants, pretty much every living thing makes these tiny proteins as their own personal bacteria fighters. They're like our natural first line of defense. So you'd think, great, let's just use those. But it's not quite that simple. They come with their own set of problems. Yeah, here's the catch. If you were to just take these natural peptides and inject them into a person, they wouldn't work very well. For one, they break down way too quickly in our bodies, and even worse, some of them can actually be toxic to our own cells. So it's clear we don't just need nature's weapon, we need a better, smarter, engineered version of it. And this is where the whole strategy just gets incredibly cool. Because instead of spending years doing endless trial and error in a wet lab, these researchers started their work inside a computer. It's a process called in silico design. And the best way to think about it is like a digital architect who's designing a new skyscraper, right? Before they ever pour concrete or lay a single brick, they build this incredibly detailed virtual model. They run simulations, they test it against earthquakes, high winds, everything you can think of. Well, these scientists did the exact same thing, but for a molecule. So here's a look at their digital design process. First, they scanned these huge public databases to find a natural AMP that looked like a promising starting point. Then, and this is key, they used AI to actually engineer an improved synthetic version. Step 3 was that digital stress test. They ran these intense simulations to predict if the new peptide would actually hold its shape and be stable. And only after a design passed that test with flying colors did they move on to the final step, actually making it in the real world. Okay, I know these graphs look pretty complex, but trust me, they're telling a very simple and really exciting story. Just look at that main line in the bottom left graph. See how it takes a sharp dive and then just flattens out? That's the computer simulation showing us that the peptide folds into a stable, powerful shape, and then it holds that shape. Right there, the simulation was basically screaming, "We've got a winner." All right, so the digital blueprint looked perfect, but now it was time for the real test. It was time to leave the world of simulations and see if this thing could actually fight in the real world. Time to put it through its paces. So test number 1 was pretty straightforward. Could this new peptide stop MRSA and E. coli from just growing in a petri dish? And the answer was a huge yes. What this table shows us is pretty incredible. At a certain concentration, the peptide stopped the bacteria from growing, and if they just doubled that concentration, it flat out killed them. The big takeaway here, it worked, and it worked powerfully against both of these superbugs. But look, Killing free-floating bacteria is one thing. That's kind of like the first level of the game. The real challenge, the final boss battle, if you will, is something called a biofilm. As the study points out, these things are somewhere between 100 and 1,000 times more resistant. It's basically this slimy, protective fortress that bacteria build around themselves to fend off any attack. First up to the plate, the MRSA fortress. And the results? I mean, just look at this. It's stunning. On the left, you see that dense, thriving MRSA biofilm before the treatment. And on the right, after the peptide was introduced, it's just gone, completely obliterated, totally dismantled. Okay, but what about the even tougher opponent? Next up was the E. coli biofilm with that extra layer of armor we talked about. And once again, it was a total knockout. On the left, you can see that thick, really organized structure of the E. coli fortress. But on the right, after the treatment, that fortress is in ruins. The peptide punched right through its defenses. So let's recap. The team has digitally designed a powerful new weapon. It can kill 2 of the world's most dangerous superbugs, even when they're hiding in their biofilm fortresses. But this all leads to one final and maybe the most critical question of all. It's a powerful weapon, but is it safe for us? I mean, what good is a superbug killer if it also kills our own healthy cells? So the final part of the study was all about safety. And here, the news was just fantastic. The peptide passed 3 crucial safety checks with flying colors. It was safe for our human cells, it didn't damage our DNA, that's what non-genotoxic means, and it didn't harm our red blood cells, which is non-hemolytic. Check, check, and check. And this image here is such a perfect way to show that DNA safety. It's from something called a comet assay. Now, look at the far left. That's a cell that's been hit with a known toxin, and you can see its damaged DNA streaming out behind it, just like the tail of a comet. But now look to the right at the cells treated with the new peptide. They're clean, they're round, they're healthy. You literally can't tell them apart from the untouched control cells. So think about it. What started as just a search through a digital database has now ended with a real-world, safe, and incredibly effective molecule that can tear down superbug fortresses. This research really is a powerful demonstration that in the war against disease, our next great weapons might not be discovered in a test tube at all. They might be designed with incredible precision inside a computer, one single atom at a time.